nervous system and the structure of the nervous system. Let's take a look at nervous tissue. What we might first thought was just nerves is made up of two really different types of cells. Neuroglia are glial cells, literally translates as nerve glue, are small cells that surround and wrap the delicate neurons. Neurons are the actual nerve cells. Those are the cells that have the electrical signals and release the chemicals that talk to other cells. Neuroglia is important to keep the neurons alive. Functionally, it's that when we talk about the nervous system, it's the neurons we think of, but the neuroglia are just as important. There are four main kinds of neuroglia within the central nervous system, and there will be equivalents of these in the peripheral nervous system. In the central nervous system, we have astrocytes, microglia cells, epidermal cells, and oligodendrocytes. The astrocytes are the most abundant, probably the most versatile. They have highly branched, they look a lot like neurons, actually, in my opinion. They cling to neurons, wrap around the synaptic endings and capillaries. And their functions include support and brace the neurons, provide some of the three-dimensional structure. They have a role in the exchange between capillaries and neurons. The changes that normally occur within plasma are too much of a change for neurons to survive. So they help to keep the fluids that neurons are in at a acceptable range of tolerance. They are important for guiding the migration of young neurons when they are developing. And with that exchange from capillaries and other things, they control the chemical environment around neurons. They do have receptors for some neurotransmitters and some nerve impulses. They have an influence on neuronal functioning, and they do participate in ways we don't fully understand in information processing in the brain. So here is your astrocyte with wraps around capillaries and around neurons. So it helps to control what gets to the neuron. The microglial cells are small, roundish, thorny processes. They touch and monitor neurons. They migrate towards injured neurons. They can phagocytize microorganisms and neuronal debris. So these are the kind of macrophages of the central nervous system. Here is one looking for microorganisms talking to neurons. Epidermal cells can be squamous or columnar, depending on where you find them. Many of them are ciliated, and those cilia beat, and that beating of the cilia helps to circulate cerebral spinal fluid, which we'll look at more closely later on. That cerebral spinal fluid is in the central cavities of the brain and spinal column. The brain and spinal column are really floating in cerebral spinal fluid, and these cells line the central cavities and help circulate that fluid around. They do form a permeable barrier, selectively permeable barrier, between the cerebral spinal fluid in the cavities and the tissues that are bathing the central nervous system cells. And here are epidermal cells with their cilia circulating cerebral spinal fluid out here. The oligodendrocytes are branch cells. Their processes wrap central nervous system nerves with the insulating lipoprotein myelin. So they form the myelin sheath in thicker nerve fibers. They provide an insulation for it. And so one oligodendrocyte will wrap around several different axons, and we'll see the purpose of this myelination. It actually speeds up the rate at which signals travel in nerves, and it's important to protect the nerves as well. And in the central nervous system, oligodendrocytes will wrap around many different axons. In the peripheral nervous system, there are two cell types that take care of the two of the functions we just talked about. Satellite cells are going to be like the astrocytes of the central nervous system. There, several satellite cells will surround a neuron cell body in the peripheral nervous system. The Schwann cells, also called neurolemmacytes, actually probably more correctly called neurolemmacytes as we try to get away from things named after anatomists, they surround all the peripheral nerve fibers and form myelin sheaths on thicker nerve fibers. So they are similar to oligodendrocytes in the central nervous system. One big difference between Schwann cells and oligodendrocytes is that a neuron will be wrapped with many different Schwann cells, whereas in the central nervous system, one oligodendrocyte can wrap around several different neurons. And in the periphery, Schwann cells are vital when a peripheral nerve is damaged. We usually say you can't replace nerves, but some of the axons can grow back under certain conditions, and one of the things they need are Schwann cells to do that. So we have macrophages already to take care of that feature of the central nervous system, and the peripheral nervous system is not surrounded with cerebral spinal fluid, so we don't need the epidermal cells. Ooh, drew a blank there for a second. Well, you're used to that. You've had me all year. Right, so neuroglia. Here is a cell that you should immediately recognize as a peripheral nerve cell. First of all, it has multiple cells surrounding the cell body because the cell bodies in the periphery are surrounded by several different satellite cells. In the axon is covered by multiple different Schwann cells. So this is a peripheral nerve surrounded by its neuroglia. That will leave us to the neurons. And neurons we're going to do in a separate set of lectures because they have some very particular cell characteristics.